MP Biomedicals offers a range of reagent kits for various types of biological and environmental samples. In this video, we will do a demonstration on how to do a sample processing using MP Biomedicals FastPrep 5G MPure-96 instrument and our new ready-to-use MagBeads FastDNA Kit for soil. Be sure to check the lysis buffer S1 to ensure there's no precipitate in the solution. Warm the solution at 55 degrees Celsius to dissolve the precipitate if there's any. Before starting DNA extraction, ensure your workbench is clean and has been decontaminated. This can be done using freshly made 10% bleach solution, clean water, and 70% ethanol. You can start by spraying your workbench, pipettes, and all equipment with freshly prepared bleach solution. Let it sit for 15 minutes before you wipe away the residual with clean gauze. Then rinse or wipe the surface with clean water and finally spray with 70% ethanol. To begin sample processing, add 100 to 500 mg of soil sample into a Lysing Matrix E tube. Ensure that there are 1/3 to 1/4 of empty space remaining in the tube. Next, add the following buffer into the sample. 980 microliters of lysis buffer S1. 120 microliters of lysis buffer S2. 10 microliters of RNA solution. Vortex the sample to mix. Take note to secure samples on the vortex by using an adapter to ensure good mixing. Fasten the mixture to FastPrep instrument. Homogenize sample for 20 to 40 seconds at speed setting of 6 m per second. The speed and time can be changed according to the types of soil samples. Alternatively, vortex the sample mixture for 5 to 10 minutes at maximum speed if FastPrep instrument is not available. If other homogenizer is used in this experiment, consult the manufacturer for the appropriate parameter. Transfer the tube to a centrifuge machine. Centrifuge the mixture at 14,000 G for 5 minutes to spin down the debris. If 14,000 G is not feasible, you could use the maximum speed available on the centrifuge machine. Transfer all of the supernatant into a clean 1.5 ml microfuge tube. Add 100 microliters of inhibitor removal MS. Mix thoroughly by inverting the tube for 20 times. Centrifuge the mixture at 14,000 G for 5 minutes to spin down the precipitate. Remove the pre-filled plate of binding buffer MS from the sealed pack and peel away the foil. Transfer 500 microliters of supernatant from sample into each well of the plate. Switch on the MPure-96 and log into your user account. Press Run. Select MBead soil. Press OK. Fill up the name for kit lot and tip lot. Insert ID for each well if needed. Place the spin tip to position 8. Press Next to move on to the next position. Place the sample mixed binding buffer MS plate to position 1. Place the pre-filled wash buffer S1 plate to position 2. Place the pre-filled wash buffer S2 plate to position 3. place the magnetic beads plate to position 5. Lastly, place the elution plate labeled DES buffer to position 6. Press Next. Ensure the eject tip plate is set to 2, and press OK. After the program ends, remove the DES buffer plate from position 6. Transfer eluted DNA to a clean 1.5 ml of microcentrifuge tube. Do note if magnetic beads are observed in eluent, centrifuge at 14,000 G for 3 to 5 minutes before transferring them to clean tube. DNA is now ready for PCR and other downstream application.